All right, so the second part of the class is going to be on the CAP system. This is also a CRP, known as CRP. So this is a protein that provides additional regulation, actually of a large number of genes, but importantly for this class and for this chapter, it regulates the LacI as well. So CAP is an acronym for catabolite activating protein, and so it also regulates the expression of the LacI, and so now we're going to see positive control, an example of positive control. So just to remember, the LAC repressor was an example of negative control. The LAC repressor was right next to the LacI, but it was a completely different mRNA that it was expressed from. CAP is another gene, and this gene is not is encoded elsewhere on the genome in a completely different location. So this is another example of a trans-acting factor. So the way it regulates the LacI is it produces this protein that can diffuse through the cell in order to regulate the LacI. So this is a way to now tie in glucose, the presence of glucose, into whether or not the LacI is expressed. And so, one of the things we talked kind of as a broad principle about for transcription, especially for eukaryotic cells, is that you can have a large number of transcription factors regulate a single locus. And so this is an example where now instead of there just being one factor, now we're making there to be two factors that regulate this operon. So there's a specific site that it binds to, and it facilitates RNA polymerase. So it makes it easier for the RNA polymerase to bind to the promoter and to start transcription. So the maximum transcription of the structural genes occurs when the repressor is bound by lactose, or actually allolactose. So it does not repress the operon expression, and then the CAP is also bound to the CAP binding site. So we are just looking at the LacI one more time, and so we have the operator site, we have the polymerase site, the promoter, and now we're adding a region within the promoter that shows where the CAP protein binds to. And so CAP, again, is an example of an allosteric regulator. So, in the same way that the LAC repressor was an allosteric regulator, where its ability to bind DNA was dependent upon binding of a second molecule. So CAP, the protein, is also an allosteric regulator. And so this regulation is opposite in the sense that it must be bound to its regulator in order to bind to the DNA. So just as a reminder, the LAC repressor, if it was bound to allolactose, it couldn't bind to DNA. But life is flexible; it can evolve any sort of mechanism that makes sense for it. And I guess not makes sense for it, but it is possible to evolve all these different sorts of mechanisms. And so, this CAP protein cannot bind to DNA unless the cofactor called cyclic AMP, cAMP, so it's a modified version of AMP, so monophosphate, so it's not ATP, which is a triphosphate. AMP indicates a monophosphate, and then it's cyclic, which just indicates that there's a reaction that creates a ring between different atoms. And so if this binds to the CAP protein, then we have this conformational change, and so this conformational change allows it to bind to the CAP binding site. And then this polymerase, the actions of this makes it easier for the polymerase to come in and start transcription. So it starts transcription unless the LAC repressor is bound to the operator, and then that prevents it. So in this case, this is a regulation of glucose versus glucose present or glucose not present. And so glucose does not bind to CAP, but the presence of glucose leads to a decrease in the cyclic AMP levels. So just remember that glucose, it has an inhibitory effect on cyclic AMP. So if there's no glucose, then cyclic AMP levels are high and CAP is bound to this region. If glucose levels are high, it decreases CAMP levels, and so CAP cannot bind as efficiently. So again, this is cyclic adenosine monophosphate. So this occurs when you have this ATP, the triphosphate, and so there's a enzyme called adenyl cyclase that comes in. It cleaves off two of the phosphate atoms to the phosphate molecules, and then this phosphate makes a covalent bond, the original oxygen, and then it makes an additional covalent bond to the 3' oxygen, the oxygen connected to the 3' carbon. And so this is why it's called cyclic AMP, because you create this cyclical ring structure on it. So again, glucose inhibits this protein, and so if glucose is around, then you have a lot less conversion, and so you have a reduced amount to cyclic AMP. So now the LAC operon can actually integrate both glucose and lactose levels to find a desired range. So this is actually a recent experiment that was done maybe 15 years ago, where they looked at the exact effects of the two cofactors on the overall expression of the LAC molecule. So just as a reminder, IPTG, that is an analog of lactose that can bind the repressor. And so if there is a high level of IPTG, then that kind of mimics a high levels of lactose. CAMP, so that has the opposite effect, so low levels of glucose in the opposite direction. So high levels of glucose indicate that there's not a lot of cyclic AMP, and then high levels of cyclic AMP indicate a low level of glucose. And so you can just see what the expression is, and so you can see the most potent places where expression occurs is when you have low levels of glucose and high levels of lactose. So this is kind of the sweet spot for this locus. So if you really want to turn on the LAC operon, you want to have high lactose and low glucose. And then as glucose levels build up, you still see a small amount of the LAC operon, but you don't have as much of it. And then if there's no lactose present, then you basically have pretty much no expression either way. And so why does the cell do that? The cell does that is because if you have both glucose and lactose within the environment, you primarily want to use the glucose first. So you want to use that glucose. It's an easier sugar to metabolize for the cells, and so they primarily dedicate their energy in order to use that source. So they still have some levels of the structural genes around that allows for the metabolism of lactose, but it's just a way to fine-tune it exactly how they want it. And so it's pretty cool. So this just kind of illustrates how you can have a very fine level of control over the expression of a single mRNA and the level of expression of an mRNA in response to two completely independent environmental sources. So now this gets to the idea of how you can have multipartite regulation of a gene. And you can continue to expand this idea in order to integrate more and more elements into the expression. And so, the more transcription factors that can bind a region, the more control you have over the expression of a gene. And so if you look into real life systems, you might find 10, 50, 100 different proteins, transcription factors can bind in the upstream region of a gene. So how does this whole process work? So again, here is the LAC operon, starts over here, and here is the promoter, and then here is the LacI gene. And so we talked before that the LAC the LAC repressor can bind to this operator in order to repress transcription. So even if this cap is bound, even if there's high cyclic AMP levels, the binding of the repressor to this region suppresses transcription. And so how does that happen? It's actually thought that the way this happens is that the LAC repressor induces a looping that prevents the polymerase and the cap from binding to it. And so it actually has multiple operator sites that it can bind to. So this is the primary operator site that we talked about, but there's other regions that have been shown to have DNA binding activity. And so it's thought that in toto, these things come in and they cause a looping between the various sites, and then as an effect, this -10, -35 region is excluded from binding.